This study is about the development of a zinc solubilizing inoculant for rice and corn, which was trademarked as Oryzinc. It was demonstrated that its use can reduce nitrogen fertilizer by 50% and zinc fertilizer by 100%. Traditional agriculture is dependent on the heavy application of chemical fertilizer to maximize yield. Unfortunately, these excess fertilizers and heavy metal soil amendments contaminate water through the leaching of nitrates and metals into the groundwater. The Oryzinc technology minimizes the use of fertilizer through the solubilization of already existing unavailable forms of zinc metals in the soil and utilization of nitrogen in the atmosphere through nitrogen fixation.
As for the methodology, we collected rhizospheric soil samples of corn, rice, and cassava from different areas, specifically in the province of Nueva Vizcaya, Tarlac, Laguna, Oriental Mindoro, Aklan, Dumaguete, and South Cotabato. We isolated zinc solubilizing microorganisms from the samples, and then we identified it using 16S rDNA sequencing. Screen the isolates further for other plant growth promoting traits such as nitrogen fixation, phosphate solubilization, and biocontrol activity against Fusarium. And then, we quantified the amount of zinc released through atomic absorption spectroscopy. And then finally, we tested it under growth room, screenhouse, and then field condition. Diversity analysis revealed the lack of diversity in zinc solubilizing isolates despite them coming from different provinces and are geographically isolated from each other. They are dominated primarily by the genus Burkholderia, Acinetobacter, Bacillus, Sphingobacterium, and they were present in 3 provinces, namely Dumaguete, Laguna, and Oriental Mindoro. Zinc solubilizing Burkholderia, on the other hand, was only found in the province of Aklan and Dumaguete. Zinc solubilization was determined using the solubilization index, which is a function of both the diameter of the bacterial colony and the halo zone it produced. The highest SI was exhibited by isolate M1, which was derived from the rhizospheric soil of rice in Mindoro, followed by isolate D3, which was derived from the rhizospheric soil of cassava in Dumaguete. SI ranges from 0.14 to 8.41. This is quite high since most solubilization indices reported for zinc solubilizing isolates are mostly under the value of 5. Characterization for other plant growth promoting characteristics revealed that 36 bacterial isolates are positive for nitrogen fixation, 13 isolates tested positive for phosphate solubilization, while 17 isolates tested positive for biocontrol activity against Fusarium. 6 isolates, on the other hand, have all these 3 biocontrol and plant growth promoting traits. Screenhouse experiment revealed that the inoculation of zinc solubilizing microorganisms can enhance the growth of rice and corn under a total lack of zinc. For corn, inoculation of the isolates has even surpassed the positive control. The most evident symptom of zinc deficiency is delayed and stunted growth. Thus, it was measured during the field experiment. The site chosen is a field in Calauan, Laguna, and soil analysis revealed that it only has 1.3 ppm available zinc. It was found that the rice plants inoculated with zinc solubilizing isolates did not differ from the positive control supplemented with complete and zinc fertilizer. Uniform height can be observed, and this is indicative of adequate zinc uptake. Grain yield likewise had the same result as that of the positive control. The grain yield ranges from 10 to 17 grams of rice produced per plant, which translates to around 3 to 4 tons per hectare. Although it is average, it was conducted in a very marginal soil with only 1.3 ppm of available zinc in the soil. Zinc quantification was conducted after grain dehulling and processing. It was found that the zinc content of rice grains increased by up to 40.97 ppm with very low maintenance and marginal soil. Another interesting observation is the increased lodging resistance as a result of adequate zinc uptake. This lodging was the result of a signal number 2 typhoon recently. Thus, inoculation of zinc solubilizing microorganisms can potentially contribute to climate-resilient crops. As for the conclusion and future directions, our team would like to make the zinc solubilizing inoculant available to farmers as soon as possible. It is currently under FPA registration under the trademark Oryzinc. Oryzinc is originally an alternative to zinc fertilizers, but it can also be a substitute for phosphate and nitrogen fertilizer as indicated by the results. The production of Oryzinc is also very cheap at around 20 pesos per 200 grams, and 5 to 6 packs are used per hectare. With all these benefits, Oryzinc contributes to poverty alleviation since it's a very cheap alternative to chemical fertilizer, climate-resilient crops through increased lodging resistance, and also malnutrition mitigation through rice grain zinc biofortification.